Hello, I'm Eric Topol, Editor-in-Chief of Medscape, and it's a real privilege for me to have Paul Offit, who is my go-to expert in vaccines and an opportune time to talk about the vaccine for COVID-19. Paul, welcome.
Thank you. Thank you, Eric.
So this is a really a critical juncture for the vaccine against SARS-CoV-2. And I know you've been intimately involved with this for quite some time. That is, at least well before there was a pandemic, and now that we're in the midst of it. Before we get into some of the nitty gritty, can you just give us your overall view of the landscape on this?
Well, it's an unprecedented moment. Never before in history have so many companies or so much money or so many different ideas for how to make a vaccine been brought to play. And so we're using every strategy that has ever been used to make a vaccine before, as well as a handful of strategies that have never been used to make a vaccine before. It's all happening at warp speed. And I think at some level, that's disconcerting. A lot of the language that surrounds this warp speed, finalist, race for a vaccine, I think scares people that either timelines are being inappropriately truncated, or worse, that safety guidelines are being ignored. But I think that we're good. I think right now, 8 companies are in the midst of phase 3 trials, large prospective placebo-controlled trials to prove efficacy, and at least to some extent, or efficacy for a certain length of time, and prove safety at least in a certain number of people. So I think it's all, right now it's all good. As long as the phase 3 trials don't get truncated, as long as the administration doesn't try and reach its hand into warp speed and pull out a couple of vaccines and oversell them because there aren't enough data yet, I think we're fine.
Yeah, to go from warp to warped, I guess, in some respect. Now, we're going to get into that. Before we do that, there's several different types of vaccines. The 2 that the U.S. has put its biggest bets on are the mRNA vaccines of Moderna and Pfizer. But obviously there's several others vaccines that you said there's 8 that are in phase 3. What's your sense, the mRNA vaccines that the U.S. has got the biggest investment emphasis have never been in people before in a vaccination program, whereas the others, at least some of the others, have. Do you think that is any worry about having a genetic vaccine?
Sure. I mean, I think when you have no commercial experience with the vaccine and you're using that as a way to try and stop a virus, this bat coronavirus that's just made its debut in the human population, has had a number of surprises, I mean, clinically and pathologically. No one would have expected that an enveloped virus, respiratory virus that is spread by small droplets, would rage in a hot and humid climate during the summer. That doesn't happen. Flu's gone. No one would have predicted that it would cause this unusual multi-inflammatory syndrome of children. That I know of no virus that does that. I think the most surprising aspect of this is that it causes vasculitis. You know, that and not because apparently these viruses, or this virus, reproduces itself in the endothelial cells that line blood vessels. I think it is a function of the immune response. I think it is a panel of cytokines and chemokines that's doing that, and as a consequence, you can have strokes, heart attacks, I mean, liver disease, lung disease, and other distal organ disease, kidney disease, because of that vasculitis. It seems to have a predilection for nursing homes. You know, influenza also kills older people, but fewer than 10% of the deaths are in nursing homes. Here, it's more than 40%.
Right.
So, so you have this difficult to characterize elusive virus that you're now about to meet with a handful of vaccine strategies with which you have no commercial experience. I think you can assume that there may be a learning curve here. And for me, I guess one of the aspects that's most disconcerting is I wish there was a little more humility from some of these companies. I mean, they're always sort of pounding their chests as to how they're going to have this vaccine. I think just within the last day, the CEO of Pfizer said, I think we can have a vaccine by the end of October. I'm sorry, are you on the Data Safety Monitoring Board? Are you seeing these things?
Really? Well, you know, we want to get into the Data Safety Monitoring Board and the Advisory Board and some of these really critical decisions that are coming up end of October, before November 3rd, that is. But before we do that, I just want to go back and forth with you a little bit on the biology you just touched on, because here we have tropism of the virus to many places wherever in the body there's ACE2 expression, as you touched on. And then we also have this immune inflammatory response to that. So it's hard to separate out, like you mentioned. You can get the virus getting into endothelial cells, but you can also get an inflammatory and cytokine reaction to that. The same as in the heart, is in the kidney, the brain. I mean, this is a really tough nut to crack, and it's obviously, there's still people dismissing this as the flu somehow or other. But moreover, the idea that we have a lot of things that we still don't understand, like, for example, the long COVID. What is your sense about long COVID?
Hard to, I mean, the sort of the long-term folks, meaning things like where you see sort of depression, joint pain, fever, low-grade fever.
Yeah. Joint pain, brain fog, you know, chest discomfort, difficulty breathing, you know, very poor exercise tolerance that goes on at least since the infections that were in March and still going on. For some people, very disabling. Unknown percent of people.
Right. I mean, I think it's a reflection of, I think, probably a couple things, probably not just one thing, but I think probably, at least in part, that there is, because of the vasculitis, that there is end organ damage in a variety of organs, and that can be somewhat chronic. This sort of scarring of the lung, I think, also can be somewhat disabling. I think we'll learn about that over time. I think there may be, you know, an ICU psychosis associated with this, that the longer that you're in an ICU, that when you come out of it, you know, sort of brain fog or depression, you know, may be part of that. So I think there may be sort of overlapping things going on. I think it's real. I don't think it's just something people are making up. I think this is a real problem, and I guess we'll learn about it as we gather more information on this unbelievable one.
Yeah, no, it's concerning. It's even in young, healthy people have mild infections to be, you know, really troubling, durable issues. All right, so now going in on the vaccine, there are the two 30,000 participants placebo-controlled trials that are expected to get their enrollment done. They're more than halfway now, the Pfizer and Moderna, by, let's say, end of this month, September. Then they, as you pointed out, they have, both of these have a 2-dose program. So after the first dose, there's a booster at a month later, and then there's weeks before you'd see the full expected immune response. So can you help me, that is by the end of October, knowing that enrollment will only finish this month at best, that this is going to give us a readout on the phase 3 trials, these two phase 3 trials?
I don't see how it's possible. I mean, I think the Moncef Slaoui, who's the sort of the head of Warp Speed, has, I think, appropriately said recently, I think on a national public radio show, that he thought best case scenario would be the end of the year, most likely the beginning of next year. I think that's a more accurate representation. I mean, as you said, you have to get 30,000 people to get dose 1 of placebo or vaccine. You have to wait a month, give 30,000 people dose 2 of placebo or vaccine, wait 2 weeks for full immunity. Then you have to hope that probably about 150 or 160 people in your placebo group get sick, not just infected, but sick. The clinical endpoint to these trials is moderate to severe disease. Plus, when Jackie Miller from Moderna presented to ACIP last week, and she was asked the question, what is the attack rate in the places where you're rolling these trials out? Because they're getting antibody studies on all these people when they enroll. So they have a sense of what the attack rate has been, and it's about 1%. So if that's true, remembering that 80% of the disease is asymptomatic, you're hoping for that 20% that's symptomatic in an area where only about 1% of people have been infected. And you don't want, you know, you're always telling people to wear masks and social distance, because 1, they may be placebo recipients, and 2, the vaccine, no vaccine is going to be 100% effective. So in many ways, you don't want them to get sick. On the other hand, you're only going to know if a certain number of them get sick. I mean, you're not asking them all to go to Sturgis, South Dakota, and hang out with maskless bikers. You know, you want them to be protected at some level.
Do you know the two programs? Are they enrolling in Brazil in high zones of infection and spread? And that rate of attack rate, has that been published anywhere, or do you know, is that just from what you've been hearing from the conferences?
Right. So the conferences, the trials are being done in the United States, South America, Africa, and so in areas that presumably are hot. So it increases your chance. Then you'll be able to get a representative number of clinically significant illnesses in your placebo group. But again, I think even best case scenario, I can't imagine that happens before the end of the year, assuming that, you know, that our goal here is the same as the same goal it is for every vaccine we make, which is a clear standard of safety and efficacy for any licensed vaccine.
Right. So now let's go through, there's a data and safety monitoring board for each of these phase 3 trials. And in that is a multidisciplinary group. You've got people who are experienced with clinical trials and biostatisticians and bioethicists and immunologists and vaccinologists, virologists. You have this big crew, they're reviewing the data, it's blinded, and they have a pre-programmed time of review. And also they have stopping rules that are defined ahead of time. This is for futility. This is for stopping for overwhelming efficacy. And the numbers, like for example, the Pfizer CEO, they gave these numbers yesterday, where it was, you know, a very small numbers of people with placebo or vaccine active, and claiming that that could, by the end of October, demonstrate adequate efficacy. These numbers seem totally out of line with what would be considered stopping rules. I mean, you're talking about giving a vaccine of any of these programs to tens of millions of people, and you're going to base that on 100 events? Well, what's your sense about what could be appropriate stopping rules that a DSMB would set up before a trial like this began?
Right. So the NIH Active Group, a group that was put together by Francis Collins to try and construct how these trials should look. There was a statistician from the University of Washington who sort of went through those data, and it was his analysis showed that you'd probably need at least 150 or 160 people in your placebo group to start to begin to say that you have a statistically significant observation, assuming that you have just a handful or fewer people in your vaccine group who were ill. That was basically his statement. And I think that's true, but you're right. I mean, you have stopping rules, but I'd like to think that's what the stopping rules are. That the stopping rule isn't that you have 10 people who are sick in your placebo group and 2 in your vaccine group, which are numbers that are far too small to make any statements about statistical significance.
Well, you know, with the CEO of Pfizer actually was citing less than 100 total events, you know, with a 50% reduction in the, you know, from the vaccine, from the placebo group. That to me, even 160 seems like a really small number for a stopping rule. You know, in such the stakes here are so big, right?
I think one could only imagine 160 stopping if you have virtually no cases in your.
Right. And we're not talking, something to clarify, because this is important. We're not talking about just a PCR positive mild infection. It has to be moderate to severe to qualify as an infection, as an event.
That's
All
right.
right. Now, the other part about this story is that not only are we getting that even one of the vaccine companies, which by the way, the DSMB isn't supposed to be reporting to the company. It's supposed to be reporting to the trialists that are testing the vaccine. How independent are these academicians that are doing the clinical trial? Does all that data go right to the company as well?
Historically, the DSMB reports to the company. That's
to the company.
Oh, they go to the company and they say that let's say we have met a point. We're at a point now where you could argue that statistically you have efficacy. You then have the option, if you want, to then stop the trial or not. And this isn't common. I mean, it's not common that a DSMB will stop a trial early, assuming there's not a safety.
No, very rare, yes.
And very rare. So then they could go to the company and say, you could stop it now if you wanted. And then the company has the option of stopping or not. Sometimes they don't stop and they continue to go on. But it's the company historically that the DSMB.
Oh, that's interesting for vaccine trials, because the trials I've been involved in, the company was out of the loop, and only after the trialists reviewed the DSMB, then there was potential discussion with the company. This is an interesting difference. So the company would know the data. Now we go to this committee that you're on, which is the Fda Advisory Committee, right? That's the committee that's meeting October 22nd, that has been called out by the commissioner of the FDA that he doesn't have to honor the recommendation. He can override that if he wants. Can you tell us about your committee? Because that's different than the DSMB, right?
Right. Well, like the DSMB, it is independent of the government. It is independent of the company. It's academicians looking at data and making recommendations. The same thing's true of the FDA Vaccine Advisory Committee. It's independent of the government. It's independent of the companies. We're paid by neither. We're just academicians. But like the Advisory Committee for Immunization Practices to the CDC, the FDA's Vaccine Advisory Committee is an advisory committee. You give advice. You can say, I think that this vaccine or these vaccines are not ready to be given to the American public. The FDA commissioner and the team that presumably the commissioner surrounds himself with then have the ability to either take that advice or not take that advice. They can always ignore that advice. I think that would be hard to do. I mean, the FDA Vaccine Advisory Committee is a public meeting. The public is seeing this meeting the same time, in real time, that we're having the meeting. And if you have people sitting around saying, I don't think we're there yet, I wouldn't get this vaccine. I just think with an already skittish American public, you would defeat yourself.
Yeah, you're talking rationally now, which is a good, that's refreshing. Now, the whole point about this, though, is we're not going to have a phase 3 trial completed by October 22nd. That was what we earlier discussed. And the only way it could be completed if there was some, stopping rule that allowed there to make the call by October 22nd, which seems to be virtually impossible based on, what we would know. Okay, you'd agree, right? Yeah. So that means that the data that are going to be discussed at your October 22nd meeting are going to be based on a midstream, still blinded, ongoing trial, right? I mean, any data that you get from these two trials that the U.S. has access to, apparently, through the companies, which I didn't even know that it was a porous between the DSMB and the companies. But that's the data that you'll have a chance to review next month?
I can't, unless the code is broken, by the DSMB, broken because they think that they've met a standard for efficacy and safety. We won't see those data. The only way we would see those data is if the DSMB said, I think we're good. It goes to the company, because the company is the one who ultimately puts that vaccine out there. I mean, it ultimately is their reputation when the vaccine is put out there. So, I mean, I would like to believe that there was no interest in the company putting a vaccine out there that's unsafe or ineffective. I mean, many of these companies have other vaccines, have international portfolios of vaccines, and this tarnishes all of them.
Yeah, a lot on the line, right?
Yeah, the FDA, I think that when we see, another option of what happens in the October meeting is we sit around the table and discuss what kind of data we're looking for to comfortably recommend the vaccine be given. I mean, do we want to see different subgroups recommended, the people who are over 65, people who have various comorbidities, et cetera. What kind of data are we looking for would be another thing to say, because, the FDA may also have other dates, this year as it moves on to see whether or not they want to sit around the table. That may not be the only thing.
Well, that gets me to, yesterday, our friend, Tony Fauci was on CNN, and he was being interviewed about the vaccine and all these prospects. I mean, it was the headlines, obviously, of New York Times and Washington Post, and had a lot of media coverage regarding this imminent emergency use authorization for a vaccine prior to a phase 3 trial completion. And so Tony basically said that he trusts the FDA, that they will make the right decision. My concern is, of course, that here they just grandstanded about convalescent plasma, no less prior to that, the EUA for hydroxychloroquine, which they had to revoke. And the data obviously hasn't supported any of those. Why would we have any sense of trust or credibility in making the call without a phase 3 trial being completed that the FDA wouldn't do this? You may not be able to comment on that, but we don't exactly have a consistent, reliable, science evidence database story that's been going on from the FDA since this pandemic started.
You're right. I mean, hydroxychloroquine should have never been approved. I mean, it didn't. There was no evidence to show that it worked. We certainly knew even early on that it could cause cardiac toxicity, arrhythmias. That was a mistake, and it was withdrawn. I feel the same way about convalescent plasma. I think that should have never been approved. I mean, those data certainly don't support its use. Both Drs. Collins and Fauci had, before that EUA was granted, said they don't think the information's there. Since it's been granted, they put out basically a paper the NIH did saying they don't think it works. And you could also make the argument that, if it's plasma, it really is plasma. The difference between plasma and serum is the fact that one, plasma contains fibrinogen. Fibrinogen is a clotting factor. You really want to give clotting factors to people who have potential clotting disorder? No. So, I mean, I think there are certainly theoretical reasons for why it would be unsafe. So that's two swings and misses. In both cases, those were therapeutic products given to people who were already sick. I'm choosing to believe, and I am an eternal optimist, that we will hold something that is about to be given to otherwise healthy people, many of whom are not likely to die from this virus, to a much higher standard.
Well, I'm glad. I like your optimism. I like to be optimistic too, and it's really good to share some of that sentiment. Now, we've been talking about efficacy, about the vaccines, but we haven't talked enough about safety. Now, the safety story here is complex because it isn't just the immediate reactions to the injections, obviously, but rather as people who got the vaccines are exposed to infections over time. Some may have an untoward immune response. It could be an antibody-dependent enhancement. It could be potentially some type of immune complex disease. Are you concerned about these other risks that have not been ascertained of low incidence that you need thousands of people to get a readout on that?
Well, it's certainly what people are considering. I mean, I think what worries them the most is a so-called vaccine-induced immunopathology. In other words,
Right.
if you look at the measles and RSV vaccines of the 1960s, those were two inactivated vaccines, which when given to people actually put them in a worse position. I mean, children who got this inactivated RSV vaccine were more likely, when then exposed to wild-type virus, to be hospitalized with pneumonia and to die than were children who never got that vaccine. The same was true with the inactivated measles vaccine, which actually was a licensed product for a little while, which caused so-called atypical measles, pneumonia, hospitalization. The reason people are concerned about those stories is that in both cases, the fear was that what had happened by inactivating those two viruses is both those viruses have fusion proteins. The way that they attach to cells, they fuse to the cell and then enter. It's not the typical attachment then brought in by endocytosis. So, and what the concern was is that essentially this pre-fusion conformation of the virus had been converted to basically a post-fusion conformation. And so you were being inoculated with something that you would not normally see when you were exposed to wild-type virus. The other thing is that at least the immunopathological response in those situations was a so-called Th2 response, which is associated with a different set of cytokines than Th1 responses. Everybody's aware of that. I mean, if you look at these papers that are coming out, even phase 1 studies, people are always looking at T helper cell responses and whether you're inducing a Th1 or Th2 response. So that's in people's minds. The second thing is regarding antibody-dependent enhancement, which certainly was the problem with the dengue vaccine. That you, instead of making neutralizing antibodies, you make binding antibodies, but not neutralizing antibodies. So those binding antibodies just serve to help the virus enter cells, so-called antibody-dependent facilitation or enhancement. You're actually helping the virus get into the cell instead of neutralizing it. Again, people are looking at neutralizing versus binding antibodies and their longevity with that in mind.
Right.
And once these vaccines are out there, because even though, let's say in best case scenario, they are testing 20,000 people, 30,000 people, that's not 20 million or 30 million people. So you're only going to know about a rare adverse event, serious adverse event post-licensure. But there are systems in place like the Vaccine Safety Data Link, which I think can pick that up. The CDC is certainly well aware of this and has systems in place to look for this once the vaccine rolls out.
I was trying to review with SARS, the original SARS, whether there was this immunopathological response to the vaccine. Are you aware that seems to be a murky area? Maybe you can straighten that out.
The animal model studies were somewhat confusing. And so I think it's hard to know to what extent that virus is going to predict this virus. They were different viruses. I mean, SARS-1, if you will, caused pretty much just exclusively moderate to severe disease. It was unusual to have asymptomatic or mildly symptomatic infection, which is why it was easier to put a moat around that virus and stop the spread. It was really came and gone within 2 years. Came in 2002, gone by 2004. MERS was a little different. It sort of came and then kind of perked along for a little bit. But those are different viruses. This is unique, this virus.
Yeah, so now let's say you start giving this vaccine ultimately whenever it gets approved, likely early next year if it's done legitimately. By phase 3 trials that have definitive evidence. But in that, there's not going to be so many people from the phase 3 that have had prior infection.
Well, they're certainly not being screened out for that. I mean, for the most part, everybody gets an antibody test when they come in, and whether they're positive or negative, they're still included.
Yes.
So that's good, because it would be impractical to do it any other way, I think.
Right. But so some people have had a prior infection in these trials. We don't know the percent, right?
Well, Jackie Miller last week when she presented, the sense was it was low, like about 1% or so.
Oh, you mean, okay. So I wondered if it was the same across the different programs. That's very interesting. Anyway, 1% is very low. When you start to immunize people where, let's say in the US, all the models suggest that we're at 13, 14% have had infections. And those people could have a different immune response than the people who are native to seeing the virus. Is that a reasonable concern?
I think it's something to keep your eyes open to. So you'll know when you look at these phase 3 trials, you'll know who was antibody positive at the beginning and who wasn't. In many ways, it's good that we're doing that. We're letting those people in because that's the way it's going to play out in a natural situation.
Right, but I guess what I'm getting at is they only represent 1%, or maybe in other trials 2%, and you know it's a low incidence event rate of severe safety issue. You're not going to see it in the trials. You only will see it when you're in the real world.
That's right.
And you know, these are the things that are, you know, concerned. What else besides these immune mediated or facilitated mechanisms are you concerned about regarding safety? I mean, obviously there's been all sorts of other things that have occurred with vaccines when they've been put out there. Is there anything else you're concerned on the safety side?
Well, so I guess I'll divide it up into different vaccines. So the mRNA vaccines, mRNA I think is a safe molecule. We all have mRNA in our body. It breaks down very quickly. But because it's such a labile molecule, it is encapsulated in this complex lipid delivery system, which is reactogenic. I think when you inoculate people with lipids, they get fever, including occasionally high fever. So there's that. And so you always wonder when you're giving a fever-inducing agent, you know, that you could either trigger or exacerbate any sort of immunological disease. So that's that. With the replication defective viruses, like the replication defective simian adenoviruses being produced by AstraZeneca, or the replication defective Ad26, which is being pursued by Johnson & Johnson, or the replication defective Ad5, which is being pursued by both the Russian and Chinese groups. So the good news is it's replication defective, which means that the virus can't reproduce itself and cause harm. The bad news is because it's replication defective, you really just get one virus per cell you get one cell per viral particle to make the protein you're interested in, which is the coronavirus spike protein. So you give a lot of virus. I mean, you're giving, I think that recent Russian paper that just came out this morning gave 100 billion virus particles. And it's around that. It's at least 10 billion virus particles. I mean, maybe you know, I feel burned because I'm at the University of Pennsylvania. But, you know, it was a replication defective adenovirus that was given to Jesse Gelsinger. You know, it was the first gene therapy.
Right.
It was a replication defective adenovirus.
I forgot about that. Everyone knows it was gene therapy, but you're giving a little more specificity. It's important, yes.
Yeah, and so you know, you worried that you could induce, you know, a massive cytokine response, which is what happened to Jesse Gelsinger, which was a massively cytokine responsive interleukin-6. I mean, now we have, you know, monoclonal antibodies directed against interleukin-6 like tocilizumab. We didn't then. So Jesse Gelsinger unfortunately died. But you always wonder that you could, when you're giving that many viral particles, that you could do that.
Okay. All right. So safety is something that we won't really know about when the trials are reporting out, let's say, you know, first quarter next year, because the safety is defined only in a relatively small number of people, especially the small number of those who've had prior infections, where you might say are the heightened concern for what they will do when they see when they have a vaccine and then they get exposed to the virus or to the vaccine per se. That's going to be interesting, I would think, right?
Well, yeah, I mean, it's just, which is true of every medical product. I mean, you can say with some confidence that you can rule out a relatively uncommon side effect in a phase 3 trial, serious side effect, but you can't rule out a rare side effect. I mean, as you go from phase 1 to phase 2 to phase 3, from 20 to 100 people to hundreds of people to tens of thousands of people, each time you do that, you reduce uncertainty, but you never eliminate uncertainty.
Right. But it's also the people. So the people that are in these trials are not very representative of Americans. That is, they're largely a white population. Underrepresented minorities are not well represented, nor are the aged or the children. Can you comment about that, the diversity of the immune response as a function of ancestry and age and other factors?
I think these products cannot be recommended, say, for someone who's greater than or equal to 65, unless there are clear safety and efficacy data in that age group.
Mm.
Similarly, I think that if we're going to be giving them to people who have certain comorbidities, the most common of which would be obesity, that you would like to see that those people are adequately represented in these trials. Children are not going to be in the initial trials. I mean, all these trials initially are everybody greater than 18. So I'm certain that these vaccines are going to start to roll out before we've ever tested them in children. And so then the question becomes, how do you test them in children when you have products which have been approved, shown to work? It's hard to do placebo-controlled trials in a child when you know you have a vaccine out there that works. So I imagine the way, I'm guessing, but I imagine the way that would play out would be as a wedge trial, meaning the same way that the Ebola vaccines played out in West Africa, which is you start to give the vaccine knowing that you're not giving it to everybody all at once. So there'll be people who get it and people who don't. And then you'll look to see sort of efficacy and safety by comparing those who got it and those who didn't. So it's not a prospective placebo-controlled trial, which means you have some confounding variables that'll enter that. But I think that's the way that would likely play out.
Right, but just to review what you said is that the highest risk people, or among the highest risk people, are those who are over age 65, and we aren't going to have data that's sufficient to address that group, perhaps at the end of the phase.
We don't have data if we don't have adequate data in the greater than 65-year-old, then the greater than 65-year-old person shouldn't get this vaccine, which would be a shame because they're the ones who are most likely to die from this infection. We have to generate those data. I can't see how anybody, Data Safety Monitoring Board or FDA Vaccine Advisory Committee or FDA decision makers would ever allow a vaccine to be recommended for that group, knowing that that's an at-risk group, knowing that's the group that's going to be getting this first tier of vaccines without having adequate data. And I feel the same way actually about minorities. I mean, you can't convince minority populations to get this vaccine, unless they are represented in these trials.
Right. So this is really interesting because there are limited entry of aged and minorities in the trials. Yet eventually, when this is rolled out, the beginning of it rolled out, the people who might benefit the most, who have the highest event rates, are going to not have data to address their needs. And when you think about the differences in the aged and in ancestries, is it because of the different immune response that might be anticipated to the vaccine?
Yeah, I mean, as a general rule, it's hard to say that if you look at vaccine safety or efficacy, that it can be divided up by gender or race or ethnic background. I mean, certainly it's true age makes a difference. But it's more, to me anyway, that you need to make sure those groups are adequately represented so that when you go to them and ask them to be vaccinated, they know that they have been studied, that because otherwise they're going to feel like they're guinea pigs, and reasonably so.
Okay, well, that gets us to trust and the public trust, and the fact that the willingness to take the vaccine, have the vaccine, is important, perhaps ultimately more important than the efficacy of the vaccine per se, because if it has terrible mistrust, we won't ever get to the level of immunity in the population. Can you comment about that issue and what, if anything, can be done to promote the trust?
Well, it's hard to promote trust now absent data. I think once we know the characteristics of these vaccines, it'll be much easier to try and explain what we know and what we don't know. Because I really do think it's going to be a matter of managing expectations. Here's a vaccine, let's say just theoretically, is 75% effective. That means that, and it'll be 75% effective against moderate to severe disease. And you know that it's been given to 20,000 people. And you know that your group has been represented. And you know that, but let's just assume that the virus is still killing 1,000 people a day and causing people to be hospitalized and suffer. It's never a matter of when you know everything. The question is, when do you know enough? When do you know enough to say that a vaccine's benefits outweigh its theoretical risk? When do you know that? And I think at that point you could say that you know that. But you have to also make sure people know that what you don't know. You don't know how long it's going to last. You're only going to know that afterwards. You don't know whether or not it causes a rare side effect. You're only going to know that afterwards. But there are systems in place to find both of those things out. And I just think we have to be honest and transparent about what we know and what we don't know.
Right. Although I think the trust factor, might be taking a hit. Let's say, in the first quarter of 2021, when we say, well, the vaccine's safety outweighs the risk. The only problem is we don't know anything about aged, underrepresented minorities, children. I mean, you basically tick off all the people that really, so that's a little bit of a concern. All right. Now, we've covered a lot, Paul. This has been, for me, fascinating to get your views, your insights. have we missed anything that's important regarding this big buzz about the imminent potential prior to phase 3 announcement, or even early stopping phase 3? Have we missed anything that's coming up end of October or before Election Day?
Well, I'll tell you the thing that keeps me up at night on this particular issue. We have two ways of stopping this virus. I think one is hygienic measures, face masks, social distancing, washing hands, and two is a vaccine. And I think with those two, we will be able to bring this virus under control. I do, but it'll take both. And what worries me on this is that if you had to pick which is the stronger of the two, I would go with hygienic measures. I mean, if I wear a mask and stand 6 feet away from you, you wear a mask and stand 6 feet away from me, the chances that I'm going to get the virus from you or you from me is about zero. You have two things going for you. One, you have a mask, which is going to prohibit the virus from small drops from traveling very far. And two, even if I didn't wear a mask, stood 6 feet away, the odds are also that you would get it. So if two things.
By the way, if you do get it, you're going to get a lower dose too, which is important. Yeah.
Like that more mild disease. The vaccine, on the other hand, if I get a vaccine and we have a vaccine that I think everybody would hope for, and it's, let's say, 75% effective against moderate to severe disease, that means 1 out of every 4 people can still get sick, including very sick. It also means probably a larger percentage than that 25% could get mild infection or asymptomatic infection, which they could still shed, and even shed to the point of contagiousness. We've been asking these trials, as they've been moving forward with these companies, to look at that, to look not only at whether they're protecting against moderate to severe disease, but to what extent they're protecting against shedding, because I think that is important to know. But people have such, I think, an unrealistic expectation of these vaccines that they see it as the panacea, as the magic to make it all go away.
The crazy stuff about sterilization immunity. I mean,
right. It's not going to happen. I just imagine that happening. I mean, I don't think the wild type viruses, human coronaviruses, do that. Why do we think this is going to happen that way?
Yeah, I mean, it isn't like we're talking about. By the way, are we missing the boat by not pursuing a intranasal vaccine for mucosal immunity?
I certainly think it's just like Flumis was. I think it's a good idea. But just to finish off this one thought, I think that if people feel that have unrealistic expectations, like that it's causing, or at least that they imagine that it's causing sterilized immunity, I've gotten the vaccine, I'm good. I don't need to wear a mask. I don't need to social distance. I can engage in higher risk activities. And then you've lost one of the important arms to bring this virus under control, arguably the more important arm, and that we won't have any impact initially because when we bring the vaccine out, when we bring the vaccine up in terms of usage, we'll do social distancing and masking down, which could end up having a sort of break-even effect.
Well, you're bringing up a critical point that I think a lot of people miss, and that is the vaccine effect could actually increase the number of people who are actually asymptomatic carriers, because they basically have protection from beyond their mucosa, but they're still got the virus in their nose and their upper respiratory tract to spread. And that's why this coupling of continued hygiene, mask and distance and these other measures are going to be important all the way through until we get out there with a very dense immunity of the population, right?
Right.
Yeah.
But it depends. I mean, there's a formula for this actually. I mean, it's if you have 75% effective vaccine against shedding, because really that's against significant shedding, then you would need to immunize about 2/3 of the American population to get the R0 to less than 1, meaning to stop spread, which is daunting. I mean, because you're talking about 2-dose vaccines, at least the ones that are initially rolling out are 2-dose vaccines. Then if you're talking about 2/3 of the population, you're talking about 400 million doses. We're not going to have that, obviously, initially when these vaccines roll out.
No, those are very practical points. Well, I don't want you to lose any more sleep. I'm losing a lot of sleep these days, but Paul, it's been a real pleasure to talk to you. I want to check in with you again, sometime in October, if that's okay, to get an update, because a lot's going on here on a daily basis. It's hard to keep up with. And you really, for years, you've been the person I look to the most to give us a readout on what the reality, the separation from whatever the hype and concerns that are out there. So thank you so much, Paul. Really appreciate it.
My pleasure, Scott.
All right.